Hi, everybody. I think you all know me by now, but I'm Ben Deverman. I'm the director of the relatively new vector engineering lab at the Stanley Center. And our group seeks to try to find gene delivery solutions to develop that will enable hypothesis testing in the models that the Stanley Center is developing. And in addition, in complementary work, we're trying to develop new technologies that will allow gene therapy to be more broadly applied in the CNS. And so in today's primer, we're going to give you an overview of the viral vectors, the AAV vectors that we work on, as well as talk about how they're used for neuroscience and a little bit about the limitations of the vectors. So I'm going to start by giving a background into how these vectors are being applied currently for gene therapy. I'm going to turn things over to Simone, who is a recent postdoc who joined the lab. And he's going to talk about the basics of AAV biology, how they're manufactured, how we purify them, and a lot of really cool new work that he's doing to standardize the vector preps so that they're more consistent from batch to batch. And then Ken Chan will talk about how AAVs are used for circuit studies and to manipulate and monitor specific cell types in the brain that have really driven a lot of the modern neuroscience. And then I will wrap up again at the end by talking about the limitations of the current technology and what we're doing in the vector engineering team to try to adjust some of these limitations. And so I'm hoping that by the end of today's talk, for those of you who don't really know what we do or what a vector engineering lab is, we will be able to answer that question and show you why it's important that we're here. And if that we're not making that clear, please just raise your hand and speak up, wait for the microphone, and we will answer your questions. All right. So AAV vectors have really become the most widely used viral vector for in vivo gene therapy. And they're ideal for this purpose primarily because they're based on non-pathogenic viruses. They're dependoviruses. They actually require co-infection with another type of virus for their life cycle. They're not replication competent on their own. Likely because of these limitations in their replication and the fact that they are non-pathogenic, our immune system doesn't respond to them as strongly as it does to some of the other viruses that were tried for gene therapy a few decades ago. And AAVs have now been used in well over 100 clinical trials, and for the most part, they've proven to be pretty safe. Not always very effective, but usually pretty safe. Another advantage of these viruses is that they can provide long-term gene expression. So if you deliver them to a terminally differentiated cell type, they can persist for presumably decades. There's actually evidence from both humans and some large animal models of expression for over 10 years. And they do this all not by integrating into the genome, but by forming a stable episomal genome in the cell. And so the other thing that makes them attractive is there's a wide range of AAVs that have been discovered in nature, as well as a number of new AAVs that are being engineered to actually have new properties. And among the naturally occurring viruses, they all have slightly different tropisms. And what I mean by that is their tropism means like their ability, what cell types they tend to favor, what organs they go to, as well as what species they might transduce really well. So there's a range of tropisms, but they're all quite overlapping, and none of them are specific to any one cell type. But we're now trying to engineer viruses that go beyond what nature has provided. So this table just shows an overview of some of the gene therapies that have been tried in the central nervous system over the past 10 to 15 years. There's a number of approaches have been tried. You can inject viruses directly into the brain or into the retina. And you can also deliver them through the vasculature and into the CSF space. And these all have advantages and disadvantages. The local injections provide high doses of virus right where you might want them, but they don't spread very well from that site, so there's some limitations there. And this is the way most of the gene therapies in the CNS have been tried so far. And many of them have shown some evidence of efficacy, but in general, they've been not as effective as we would like because you don't get much spread of the virus from the injection site. The going IV has kind of the opposite problem. You can get spread to all parts of the brain, but the efficiency is not very high. And the CSF space is kind of the same way. Within these trials, there have been a few that have actually progressed to approved drugs. So the first was a couple years ago, AAV2-based therapy for Leber congenital amaurosis, a form of inherited blindness, was approved both first in the US and then in Europe. And this is really allowing kids and young people who were losing their vision to maintain and improve their vision, and it's really quite remarkable. The other recently approved gene therapy that was approved less than a year ago is for spinal muscular atrophy, so a severe form of spinal muscular atrophy. So these kids generally are born with very little motor control, and it declines from there if they're not treated. But with this gene therapy that is basically taking an AAV9 vector, injecting it intravenously into these patients at very high dose, some of these kids are actually walking and talking and becoming normal, having a much more normal life, and it's really, really exciting. If you want to see, all of the kind of thing that we're doing to try to take genetics and apply it to new therapies and see this in action, Avexis has, the company that did this trial, has some videos online where you can see these families and these kids, and it's really quite remarkable and inspiring. So the goal, though, is to really try to take this even farther and start applying gene therapy to a wide range of diseases. And there are a number of clinical trials going on now that look pretty promising. There are treatments for Duchenne's muscular dystrophy as well as limb-girdle muscular dystrophy, which there were some recent results came out from this. There have been trials for both hemophilia type A and B for treating bleeding disorders, as well as a pretty encouraging trial for trying to treat Parkinson's disease by delivering the AADC gene to the striatum, where normally the dopamine terminals are releasing dopamine. When you lose these cells, this is by putting this enzyme in, they make the cells that normally respond to the dopamine and make them able to make their own dopamine from L-DOPA. So most of the Parkinson's patients eventually become resistant to L-DOPA, and this allows them to be sensitive again to it. But really the goal will be to try to apply this broadly to a wide range of both monogenetic diseases, potentially more complicated diseases, as well as even infections. And this is there's a lot of excitement in this field right now, but really what's holding this field back in large part is the delivery of the genes. I mean, we know often what gene we want to deliver. Sometimes there are packaging constraints, so you can't deliver a very large gene, but for the most part, a lot of these diseases could be treatable if we could just deliver the gene to where it needs to go. And so our group and others are trying to engineer viruses to make them better carriers of these genes. So we do this by working at two levels. We engineer the capsid, the protein code of the virus that protects the genome and delivers it to where you want it to be delivered. We use protein engineering and in vivo and in vitro screens and selections to try to find viruses that are better than what nature has given us. We also are engineering the genome, so we use regulatory elements to try to control the expression of the transgene that you are trying to express. This could be both enhancers and promoter sequences, as well as 3' UTR sequences like microRNA binding sites that could be used to turn off expression in certain off-target cell types. And I'll tell you a little bit later about some of the work that we're now doing in collaboration with Gordon and Jordan about trying to develop some high throughput assays to identify enhancers that can provide cell type specific expression. Before I do, though, I want to turn things over to Simone. He's going to talk about the basic AAV biology and how we make the viruses.
All right. Thanks, Ben. Hi, everyone. So yeah, if you look at that, it actually gives you the different steps that are required for AAV transduction. So in a nutshell, it starts with binding at the cell surface, followed by endocytosis. It traffics from the plasma membrane to the perinuclear area, so through an endosome. In the trans-Golgi network, you actually have a pH-induced conformational change that leads to externalization of one capsid domains that contains a phospholipase domain. You're going to have endosomal escape followed by nuclear entry, genome release, stabilization, and transgene expression. So if you zoom in on one of those particles, what you can see is this capsid. It's a non-enveloped virus that measures approximately 25 nanometers in diameter, and its capsid follows a T=1 icosahedral symmetry. So you have three main symmetry axes, the two-fold right here, three-fold with the three spikes, as well as the five-fold pore that you can see on that picture at the top. If you look inside, you'll find 4.7 kb ssDNA genome that contains two main genes, rep and cap, flanked by inverted terminal repeats. So rep encodes four multifunctional proteins, rep 78, 68, 52, and 40, that are driven by P5 and the P19 promoters, respectively. They are necessary for genome replication, rep cap gene expression, as well as genome packaging. Cap. First, it encodes the three structural proteins, VP1, 2, and 3, that form that viral capsid right here. They are driven by the P40 promoter. You need 60 of them to form that viral capsid, and they are present at the ratio 1 to 1 to 10, approximately. So you need approximately 50 VP3, 10 VP1, and 10 VP2 to make one of those capsids. Cap also encodes the assembly activating protein that was shown to be required for assembly of most naturally occurring serotypes, as well as synthetic ones. And finally, the MAP, membrane-associated accessory protein, that was discovered this year by the Church Lab and whose role is still kind of unclear, but was shown that it plays a very important role in virus production. Finally, the ITRs, so they flank the viral genome. They are involved in genome replication, packaging, and regulation of gene expression as well. So how do you produce those? Well, you can just take mammalian cells and you transfect them with a cocktail of two plasmids. The first one contains your viral genome, and the second one contains helper genes that are necessary for virus production. So it can come from the adenovirus or herpes. Then, when you actually take that viral genome and you replace it with a therapeutic transgene, you can also supplement your mix with a rep and cap in trans, so in another plasmid, in the absence of ITRs. If you do that, then your transgene is going to be packaged, and you can further use that vector for your experiments or for gene therapy. So how do we actually, once you produce them, you also need to purify them prior to running experiments like gene delivery experiments. So you can take your producing cells, you usually lyse them, clarify them, and then you have two main purification techniques that actually can be used in combination and that we have both been using at the lab. First one's chromatography, can be affinity or ion exchange. And the second one that's being used by most vector cores relies on density gradient ultracentrifugation. So it can be iodexanol or cesium chloride, for instance. And you're going to separate particles based on their density this time. After that, you buffer exchange, concentrate them, and further characterize them. So I'll just like to add here that both techniques have their advantages and drawbacks. If you want to produce many preps in a single run, in a few days, you're probably going to go for density gradient ultracentrifugation. It's also quite serotype independent. But if you want to scale up, you found a variant of interest, then using this becomes challenging. So you actually probably would like to switch to a column-based technique. In terms of quality control, here I would like to make a point that titers actually matter a lot. You can order AAVs from a bunch of different cores, and if you can reproduce your data from batch to batch, the culprit is most likely going to be the titers. So every facility has its own titration method. And if your titers are not correct, when you want, for instance, to run a comparative analysis of 3 different serotypes with a new variant that you identified, most of your conclusions are most likely not going to be reliable. So if you can do binding assay or transduction, assess transduction efficiency or antibody neutralization, if your dose, if your titers are not right, this is not going to be right either. So batch-to-batch consistency is very important. And since every lab has its own protocol, then it also can become challenging to actually reproduce experiments between different facilities. So how do you quantify those? How do you titer your viruses? Well, you usually just like DNA treat them to get rid of whatever is outside of the capsid. You extract those genomes and you titer them by qPCR or ddPCR. But now, one lab has shown that if you take like a single prep and you distribute it in like 16 different labs and you ask them, can you please titer that with this standard protocol that I'm giving to you, you see that you have up to 70-fold differences between them, which gives you an idea of why titration is kind of challenging. So to overcome that issue, what we did was to follow the FDA recommendation that consists in just using reference standard material that you can order from ATCC. You produce your own prep and you actually calibrate all your quality control assays using that reference standard material right here. So you can further use that standard here that you calibrated and that you have available at the lab to assess user-to-user variability, batch-to-batch consistency, and try to reproduce your experiments as well as possible between the different laboratories that are working on the same project. We make sure those are pure by SDS page, so those are 3AV8, 3AV PHP EB preps, as well as the reference standard that we produced for the lab. You can very clearly see VP1, 2, and 3 with a pretty clear purity profile. We look at the presence of aggregates by dynamic light scattering as well. And finally, we also try to assess the ratios of like empty and full. If you produce AAV preps, most likely, I would say more than 80% of your particles are depending on the production system, but with the triple transfection, you're gonna get up to like 90% of empty particles. And you wanna get rid of those. They don't contain any transgenes, so they are not gonna be very helpful for your experiment. They can actually introduce some bias in your experiment. So, as many others, we have like implemented some density gradient ultracentrifugation protocol to separate empty and full, following, for instance, chromatography. And we also have the ability to assess the ratio of empty and full by analytical ultracentrifugation. So what I'm showing you here is a sedimentation profile. You have your sedimentation that's being followed at different time points. And I'm showing you the absorbance as a function of the distance from your center of rotation in your analytical ultracentrifuge. So when you don't separate empty and full, you see that you have two populations that form those two transitions right here. That's what you get for the full one and the empty particles. So you can further derive sedimentation distribution for those. And indeed, you see that using one of the protocol we designed at the lab, we are actually capable of separating empty and full quite efficiently. And that's so the kind of ratios that you actually get in the absence of separation, about 27% full and 72% empty. So I try to give you basic concepts involving AV biology, just show you how we were producing them, purifying them, characterize them, to ensure reproducible experiments. Now Ken is going to show you what you can actually use those vectors for your experiments and your research.
Okay, so as Ben said earlier, AAVs are widely used in neuroscience, and it's because they can enable the delivery of a wide variety of genetically encodable tools. You can deliver genetically encodable tools that will allow you to manipulate cell states, such as designer receptors that can respond to synthetic ligands in order to activate or inhibit neuronal firing. Or you can deliver light-sensitive proteins called opsins that when you activate with light, they will channel or pump ions across the cell in order to excite or inhibit the cell. And these genetic tools enable techniques called chemogenetics and optogenetics, and neuroscientists use these tools in order to probe how biological activity can relate to behavior. AVs can be used to deliver genetically encodable sensors. So there are sensors that can sense the presence of calcium or certain neuromodulators, or even changes in electrical activity in a cell. AAVs are commonly used to deliver fluorescent proteins in order to help characterize cells or circuitry of the brain. In addition, you can even use them to deliver genetically encodable enzymes that can be used to manipulate DNA, such as recombinases that can conditionally turn on, off, or knock out a gene, or you can deliver base editors that can actually physically change the genome sequence. And finally, you can use AAVs to model diseases by delivering a disease-causing gene, or you can even treat diseases by delivering a therapeutic gene. When you use AAVs together with the vast amount of existing transgenic mice lines that express Cre under a specific cell type, they become very powerful tools in neuroscience. And this is because it allows you to manipulate or monitor specific cell types and dissect out how they function normally, and then how they may differ during diseased states. And so it will inform you of how to design better treatments for disorders of the brain. And this cell type specific control is done by encoding the AAV genome in such a way where it can't express its genetic tool unless Cre recombinase is present. And in this intersectional strategy, Cre recombinase is only present in specific transgenic mouse lines. To give an example of how powerful this intersectional strategy can be, this is a study from David Anderson's lab at Caltech. So what they're doing is they're using an AAV to deliver a Cre-dependent excitatory opsin into the ventromedial hypothalamus of SF1 positive Cre mice. And when they activate these SF1 cells in the ventromedial hypothalamus with light through a fiber optic cable that's been implanted in the ventromedial hypothalamus, you'll see that activation of these cells is sufficient enough to evoke a very strong fight or flight response in the mouse. So the light is turned on at the very beginning of the video, and then when it's off, the mouse persists in this defensive emotional state. So it's free because it's freezing and it has its tail up. But eventually, the mouse does go back to more normal behavior. You can also use AAVs to deliver genetically encodable toxins. So in the same study, the researchers used AAVs to deliver a genetically encodable toxins to kill off these SF1 cells to show that they're necessary for predatory avoidance. So in the top video, what they're doing is they're using an AAV to deliver a control gene that doesn't ablate these SF1 cells in the ventromedial hypothalamus. And you'll see that the mouse's innate behavior when placed in a cage with a rat is he's very hesitant when he's exploring around. So he'll go to the corner and then he'll, hesitantly go explore the rat and then back up a little bit. But if you use an AAV to deliver a genetically encodable toxin to kill these cells, you'll see that the behavior is quite opposite from the video above, and that the mouse is more exploratory towards the predator. So even towards the end of the video, he'll try to get into the cage by climbing up it. In addition to being able to deliver genetic tools that can manipulate cells, you can deliver genetic tools to monitor their cells. So AAVs can be used to deliver sensors to monitor neuronal activity. And AAVs are commonly used to do this because they can control the density of the amount of cells that are expressing your sensor, something that's not possible to do with transgenic models alone. And this is important because if you express your sensor at too high enough of a density and you have too many cells expressing your sensor, then it becomes very hard to read out single cell activity. But if you use AAVs to deliver a low dose of your sensor, then you get sparse delivery of your sensor in order to read out single cell activity. So I'm going to show a few examples of AAVs delivering these genetically encodable sensors. So a very common sensor is called GCamp, and it fluoresces in the presence of calcium. And it can be used as a proxy to read out when cells are being excited. So in this experiment, the researchers are delivering this GCamp sensor into the V1 column, and they're monitoring cells during visual stimuli. Another type of sensor that AAVs can deliver are genetically encodable voltage sensors. So in the middle here, Quasar 3, which is a voltage sensor developed by Adam Cohen's lab at Harvard, can sense membrane potentials and relate changes in membrane potential in the form of fluorescence. So what the researchers are doing here in this figure is they're simultaneously measuring fluorescence of a cell while patch clamped onto the cell. And you'll see that changes in fluorescence in red correlate with the changes in electrical activity of the cell seen in the traces in black. And finally, there are genetically encodable sensors that can sense neuromodulators, the presence of neuromodulators, such as dopamine, norepinephrine, acetylcholine, GABA, just to name a few. So what we're looking at here is a fluorescent sensor that detects the presence of dopamine and fluoresces. So in this experiment, this dopamine sensor is called D-Light1. What they do is they inject D-Light1 into the nuclear accumbens. And dopaminergic neurons in the VTA of the brain project to this area. So they're going to use optogenetic stimulation of dopaminergic neurons in the VTA and monitor fluorescence in the nuclear accumbens. And you can see on the graph on the right that the more optogenetic stimulation, the more bulk fluorescence that they're able to read out in the nuclear accumbens. AAVs can be delivered. AAVs can deliver a Cre-dependent fluorescent protein to assess newly generated Cre transgenic mouse lines. So here, in collaboration with Ling Ling and Jen Pan's group, they're interested in the CAV3.3 gene because it's the candidate gene for schizophrenia. So what Ling Ling did was she delivered with AAV PHP-EB encoding a Cre-dependent fluorescent protein to assess a new transgenic mouse line that she generated. So on the very right is the mouse line that she generated compared to the in situ data at the Allen Brain Institute. And on the left is an area called the TRN that they're interested in. And so this experiment took about a few weeks with the use of an AAV, but in order to, without them, if you wanted to report the Cre activity in your transgenic mouse line, you would need to cross it to a mouse reporter line. And this would take about a few months. And the other advantage of using AAVs to read out the Cre activity of your transgenic line is it actually detects whether Cre whether Cre is actually active during the time of the experiment. But if you use a mouse reporter line to assess your Cre transgenic mouse line, what the mouse reporter does is it marks the Cre positive cells early on in development, and that mark will persist on into adulthood, but it won't tell you whether or not your Cre is actually still active in adults. So understanding the connections of the brain is important because it allows us to understand how information is relayed across the brain. So AAVs can be used to label the input neurons to specific brain regions. And the reason why they can do this is because they can be uptaken by the axon terminal and transported back retrograde to the cell body for it to express its gene. So what we're looking at here is a variant of AAV2 called AAV2 retro that's been engineered to have enhanced retrograde transduction properties. And so in this experiment, what they're doing is they're using AAV2 retro to encode and deliver a Cre-dependent fluorescent reporter called TD Tomato into the basopontine nuclei of RBP4 transgenic mouse line. And so on here, what they're showing is that they're able to label layer 5 neurons that input into the basopontine nuclei. And it should be noted that a lot of labs are now using AV2 retro, but its retrograde activity doesn't work across all circuits of the brain, so care should be taken before using it. But what about tracing inputs? What about tracing where neurons output? So AAVs are capable of doing anterior grade tracing, meaning that it can actually fill the cell with fluorescent proteins because it could offer high expression in a cell. So the Allen Brain Institute has utilized AAVs to perform anterior grade tracing, and so they're systematically tracing where the projections are going of specific classes of neurons across the brain. And so to give an example of what's being shown here, so on the very top, they're injecting different areas of the cortex, and then they're looking at where the axons of passage of the terminals are ending up across the brain. So they'll look at areas like the striatum, the thalamus, and they'll look at the zona incerta. And so they've offered this resource online where you can actually search specific classes of cells and go look at these images, and they offer a way to actually look at the whole brain in 3D, so the resource is really cool. So finally, interestingly, the AAVs may be capable of low efficiency anterior grade transsynaptic transport. So transsynaptic transport means that when you deliver an AAV, it's able to get into a cell and then infect the downstream cell. And so there have been some reports that cells are being labeled away from the injection site where the axons terminals are. So in 2016, a group from USC showed that you can utilize this low efficiency anterior grade transsynaptic transport for tracing. So what they've done here is they either encoded using AV1 to encode GFP or AV1 to encode Cre. And so when they deliver GFP when they deliver this AV1 into the visual cortex, and they look at where the axon terminals are, they don't see any cell bodies. But what's really interesting is if they deliver AV1 into the visual cortex, encoding Cre, and they look at where the axon terminals are, there are cell bodies that are labeled in a reporter mouse line. And so what this Cre is doing is it's turning on cells around the axon terminals. And this could be because you need a very small amount of Cre to actually cause a permanent change in the reporter line to detect, to mark those cells. And they also show that only certain AAVs are capable of doing this anterior grade transsynaptic transport, so AV1 and AV9. Okay, so AAVs have been really transformative in neuroscience when you combine them with transgenic mouse models. And so they offer cell type specificity, so that they allow you to manipulate and monitor specific cells when using this intersectional strategy. In addition, they allow you to have spatial control in your experiment because you can choose where you can deliver your AAV into the brain. And they can provide connection specificity because of their retrograde and anterograde properties. And similar genetic tools and approaches are available in fruit flies. And however, using these types of genetic tools and approaches in a non-transgenic animal to study more complex behaviors or functions still remains very challenging. So I'll hand it back over to Ben, who'll talk about how we can address these things.
All right, thank you, Ken. So yeah, Ken has now shown you all the cool things that you can do with AAVs, and I have to tell you all the limitations of the AAVs and why this is more difficult to apply to the animal models and cellular models that the Stanley Center is supporting and interested in developing. So, okay, so I'll start off just by kind of restating what Ken has talked about. So one of the things that we're interested in doing is developing tools that allow you to get cell type specific gene expression in non-transgenic animals. So the goal here would be to, instead of crossing or putting a virus with a Cree-dependent expression cassette into a Cree animal here, now we need to do this without the Cree animals. We need to figure out how we can do this with just the viruses alone. So I'll talk a little bit about that. I'm going to talk about how we are trying to engineer AAV capsids to cross the blood-brain barrier and as a way of both helping with hypothesis testing and potentially develop gene therapies for both neurodevelopmental disorders and neurodegenerative disorders. We're also interested in trying to target some currently very difficult populations of cells to target, such as microglia. And we want to be able to improve the tools for studies of circuits. So as Ken mentioned, there are AVs, the AV2 retro especially, that are able to undergo retrograde transport from terminals back to the cell body and transduce these cells when you inject them at a distance. But this doesn't work across all circuits, and there's still a need for some tools to complement this vector and work in other circuits where it doesn't. As Ken talked about the AV1 Cree method of trying to get anterograde transduction and use that for anterograde circuit tracing, I think there's a big need for tools that will do this more efficiently. So right now, the only tool that's really available for efficient anterograde tracing is HSV. And the problem with HSV is that it's toxic to cells within about a day or so. And so the neurons all die. You infect them, this virus is actually replicating. And so you can, if you time things carefully, you can do circuit studies. The problem is that you can't use these for functional studies because you're going to kill the cells really quickly. So you can't use them to deliver opsins or sensors that you might want to use for longer term studies. And so I think the fact that AV1, when it expresses Cree, has shown some activity at doing this transsynaptic transport, I think that leaves room for engineering to actually try to improve that. It's something that I wasn't sure whether that would ever work until I saw that data, and I've heard from a few others that it actually does work. Another thing that we're interested in doing is looking at how genetic variability or genetic variation within the human population will actually affect the ability of AAVs and other viral vectors to transduce cells from these individuals. So are there genotypes that are more or less resistant to AAV transduction? And finally, we're also interested in trying to improve all of our library approaches that we use for screening, and one of the things that we're doing is trying to use machine learning to make better libraries and predict capsid fitness, and I'll talk a little bit about that too. So first, we've gone through all of this, Ken has gone through all of this on how AVs can be used to deliver a wide variety of things and to deliver them to specific cell types when used in Cree transgenic animals. We're trying to figure out how to do this across models that the Stanley Center is interested in developing without the use of transgenic animals or systems. There's some data that this is possible, so there are a number of gene regulatory elements that you can take from genes that are expressed in specific cell types. Some of them are small enough you can fit them into an AAV, and you can restrict expression to certain cell types. So these are all examples of places where we've taken our PHP-EB virus that transduces the entire brain, put in a gene regulatory element that restricts expression to a specific type of cell, and then looked across the brain for expression. So we can take a region of the tyrosine hydroxylase gene, which is a gene necessary for dopamine production, and we can get expression that is quite specific and efficient within the SNC and VTA, where the dopaminergic neurons are. Same thing for serotonergic neurons, FEB. Regulatory elements can drive expression in the dorsal Raphe nuclei here, in serotonergic neurons, here labeled with 5-HT. Jordan and Gord actually developed this, the DLX, as a gene regulatory element, an enhancer within the genes between DLX 5 and 6. They developed this enhancer as a way to target forebrain interneuron populations, or GABAergic neurons specifically. I'm sorry, everything is faded out and you probably can't see anything. You're just gonna have to trust me. There are lots of little red dots here, and each one is an inhibitory neuron. This is both quite specific and quite efficient at targeting these cells across the forebrain. Another example here that's probably a little bit easier to see is you can use a PCP2 promoter to drive expression specifically in Purkinje cells in the cerebellum. And again, all of these are cases where the virus went everywhere throughout the brain, but you only get gene expression in certain cell populations. So, but the problem is that there aren't that many of these known. So I showed you a few examples. I couldn't make this list that long. There aren't that many gene regulatory elements that have been shown to provide specific expression to the cell types of interest. So Gord and Jordan have been actively trying to find more of these and specifically focused it on interneurons, their favorite cell type. So the general approach is that you look at RNA-seq data, single-cell data, attack-seq data, and find evidence of where enhancers are within these, and then actually take these enhancers, clone them, put them into a AAV vector, put them into the PHPB virus, and then test them one by one in mice. And this is working, and they've found some really interesting candidates. They've also used this to find DLX, but it's slow, and it takes, it's resource intensive, and it takes a long time. And so we are now collaborating on this to try to speed up this process. So we want to take some of our expertise in doing high throughput screening to develop a system where we can actually test hundreds, if not 1,000 or so of these in individual animals at one time. And so we just got word that we got funding to do this from the NIH Brain Initiative, which we're really excited about. So we are going to start working on this full steam ahead soon. So another thing that we have been putting a lot of resources and time into is trying to find ways of delivering genes throughout the entire CNS by crossing the blood-brain barrier. This would obviously have a lot of applications both for testing hypotheses and potentially developing gene therapies around treating neurodevelopmental disorders. If you want to treat something like Rett syndrome or SHANK3 mutation or deletion, can you do this with a gene therapy? If so, when do you need to treat? They're developmental. They're probably, the earlier you do it, the better the outcome is going to be. There are other applications like for spinal muscular atrophy type 2, which is a little bit less severe and going into older patients. The IV approach with AV9 that has been working with SMA type 1 is probably not going to work for this unless the kids were diagnosed really early. You could also use a CNS-wide gene therapy to try to treat things like Huntington's disease or Friedreich's ataxia or the large number of lysosomal storage diseases that are available. And so we've been working on this for a while. And again, I'm sorry you can't see this very well, but this, we've started with the AV9 virus that is now part of the approved drug for spinal muscular atrophy and engineered into the PHPB family of viruses that are much more efficient at crossing the blood-brain barrier in mice. And we've done this, just to go over it briefly, we've done this using a system that we call CREATE, which is basically, again, leveraging the power of all the Cre transgenic lines that are available. We can make a library of viruses that has a Cre-dependent switch in it that allows us to flag the viruses that get into a specific cell type of interest. And so within this system, we can build libraries of millions of different viruses that have 7-mer peptide insertions on their surface and select for the ones that get into specific cell types. And this is how we identified the PHPB viruses. But as you can imagine, as we've already talked about, this is not an approach that is going to work very well in non-transgenic animals. If we want to try to find viruses that work in humans or non-human primates, there aren't Cre lines available, obviously, to do this. So since arriving at the Broad, we have now identified the mechanism by which our PHPB viruses can actually get across the blood-brain barrier by collaborating with the HALE team to actually do some cool mouse genetics. We've identified this protein Ly6A or SCA1, as it's also known, as a receptor for these viruses, and this gives us some clues on how we could start tackling this problem in humans. The problem is that this gene is mouse specific, so it's not, we can't just try to target a slightly different mutation, or not a mutation, but a slightly different gene variant of this in humans. So we are now taking a variety of approaches to try to select for viruses that would function like a PHBB type virus, but work in humans and non-human primates. And so how do we plan to do this? So we're going to stick with some of the things that we know work. For instance, we think that putting in short random insertions into the capsid seems to work quite well. We've used this to identify a variety of variants that have different properties. We don't see any reason to largely change our focus on that. But what we would like to do is to try to make this process a little more efficient. So we're collaborating with Fatma and Casper's lab to actually try to apply machine learning to make our libraries even better. And so the issue with any protein engineering application, especially AAV capsids, is that the sequence space that you can explore when you're engineering these proteins is huge. It's more, there's more possibilities than there are stars in the universe by many, many, many orders of magnitude. So we want to try to make as many, we want to change as much as we can about these capsids, but that we have to balance that with the fact that the more we change, the larger the fraction is that we break. So we're going to break almost all of them if we do too much at once. And so we're trying to use machine learning to try to help us with that balance and actually be able to make more mutations within the AV capsid and try to develop things that work even more efficiently. And so what we do is we take our libraries of data, we show those, and we use those to train a machine learning model, and then try to use that model to then predict what other sequences might actually make a functional capsid versus what is actually going to break the capsid. So another thing that we really have to solve, and I think this is our major challenge. We know how to make pretty good libraries. We're having some success with machine learning. The main problem is how do we actually screen for variants that are going to work in humans? And this is the tricky part. And so we could just say, well, non-human primates are closer to humans. Let's just do some selections in non-human primates. But the challenge is that we don't know if we're going to run into the same problem we did in mice, that we end up finding a virus that is species specific. So one of the approaches we're taking at least is to try to screen viruses across many species. So where we take the same library and we put them into a variety of species and try to find something that is going to work through a conserved mechanism. And so part of this is funded by the Stanley Center, and part of the work that we're doing is actually funded now by the NIH. And we're doing this in collaboration with Guoping Feng's lab to help us with some marmoset selections. And finally, one other key component to actually doing these selections is figuring out the best way to read out what are the best viruses. And so what we had done before, as I mentioned, is we used a Cree-based method to not just look for any virus that might get into or make contact with our cell type of interest, but we wanted to select for the viruses that are actually functionally transducing the cells. So our Cree-based approach looked for this step. So we were looking at the DNA level, at the genomes that had become double-stranded DNA and were stabilized in the nucleus. And so that's, I think, what provided a lot of extra selective pressure in our screen to find functional variants. But again, this is not really appropriate for doing experiments outside of the mouse. So we've modified our methods, and now we are using a RNA-based method where we actually read out capsids at the RNA level and use this as a selection. So we're, again, really focused on finding the AAVs that are functionally transducing cells and turning on gene expression. And so this is working quite well, and we're now using this across a variety of different selections. Okay, so one other thing that we are interested in doing is trying to solve another problem. So what we would really like to do is study microglia, of course. I mean, there's the C4 connection that the Stanley Center is very interested in, and just the fact that microglia seem to be showing an ever more important role in a variety of CNS disorders. And so the goal would be to be able to study these cells and manipulate them genetically, just like we can do neurons and astrocytes and other cells. The problem is that these cells don't get transduced by the AVs very well. If they did, we would be able to do things like look at how we could try to block the activation of microglia into specific states, or try to activate them. We could express or turn or knock down specific genes in microglia to ask questions about how those potentially disease-associated alleles affect the function of the cells. And so we're working with Beth Stevens' group and Matt Johnson. We co-hired a postdoc, Sasha Jarab, who is doing excellent work trying to solve some of these problems. But as I mentioned, the current AVs don't seem to do this very well. So this is actually an image just as an example of what the brain looks like. Again, I'm sorry you can't actually see both colors very well, but the green dots are all cells that are transduced by our PHP-B virus. The magenta, which you probably can't see at all, are microglia. The whole point, though, is that all the places where there are microglia, there's no GFP. So despite the fact that most of the brain is a field of green, we don't see any transduction of microglia. This is not just from our PHP-B virus. We and others have had a lot of trouble transducing these cells with a wide range of AAVs. So the question is why is this? And I think we are trying to take an approach where we study this more systematically. So we are doing selections using some of our new techniques to try to find viruses that transduce microglia very well without necessarily having to understand what bottlenecks we're solving. But we're also taking an approach where we want to stepwise look at different stages of the virus transduction of these cells. So do they internalize as well as they do in other cells? If they do internalize, then what happens to them next? Microglia are kind of designed to take up particles. I would think that they're probably able to take up AAVs. There's probably some intracellular step that might be the problem. Maybe they're just sent to the lysosome instead of going through their productive transduction trafficking mechanism where they go back through the Golgi and then to the nucleus. Maybe there are innate immune responses to the genomes or the capsids in these cells that prevent their transduction. And maybe there's mechanisms where gene expression is actually silenced and prevented in these cells. So we don't know. We're trying to look at this systematically. So we're also interested in trying to understand the variability that happens after AAV transduction. So if you look in things like the hemophilia gene trials, for instance, where they can actually have a quantitative measure of transduction by looking for factor 8 or factor 9 in the blood, and they can assess this across patients. There's a huge amount of variability, and some of this may be explained by pre-existing antibodies or immune responses against the virus, but I suspect there's also some role of the genome or the genotype, and people are probably different in how well our cells are going to be transduced by AVs. So we're interested in trying to study this together with Kevin Egan's group. So we are interested in taking our AAV vectors, probably several of them, putting them on some of the villages that they're developing after they've been differentiated into some relevant cell types, CNS cell types, and then separating the cells based on how well they're transduced by the virus, and then trying to look at the genotypes of these different individuals. And this is work that's being led by Alina Chen, who joined my group earlier this year. And so I'll just summarize by saying that our goals are really, in the Stanley Center, our goals are really to try to help scientists here actualize their therapies and model psychiatric disorders. And we've established collaborations with many of you in this room, and in projects that I haven't had time to talk about. And if you have further ideas for collaboration, please contact us. We're always open to discuss things and excited to start new projects. We spent this last year really trying to build core technologies that will allow us to be successful in the years to come, and build these technologies that will allow us to do gene therapy and support basic neuroscience studies. In this year, we've uncovered some new ways that AAVs can actually get across the blood-brain barrier and actually starting to understand the mechanism of this. We've refined methods to reliably and robustly produce AAVs in our AAV libraries, and we've innovated some new approach to screen for functional AAVs in any model system. So no longer just it's restricted to mice, but in any model system, including human cells. And finally, we are harnessing machine learning to try to further explore the AAV fitness space. And so, and also to try to understand how gene therapies may, how gene therapies could ultimately benefit a wide range of genotypes. And so with that, I'll end, and I hope that we've been successful in trying to answer any questions that you might have had about what a vector engineering lab is and why the Stanley Center needs one. This is the vector engineering team. I think we're all open to answering any of your questions anytime, and so please come to us. I did have a slide that I wanted to show that was completely full and very small type of everyone who we are collaborating with, who has helped us out in some way since we've gotten here, but I realized that I'd probably end up forgetting somebody, so it was better to thank everyone as a group. It's been a really wonderful place to set up a lab, and I'm super excited about working with everyone in the years to come. So, thank you.
Thank you, Ben. Does anyone have any questions?
Thank you. Maybe this question is to Ken. I'm very much interested in this AAV1 anterograde tracing. Is this specifically monosynaptic or multiple synaptic anterograde transduction?
So in the study, they don't show whether it's like a single synaptic jump or multiple synaptic jump. I think they only show that, when they inject one upstream neuron, that it can only label one downstream neuron, but I don't think they show.
I assume the downstream one is a Cree-dependent reporter line. Is that true?
Yes. So
Okay.
so that's injected exactly. So that AV1 Cree is injected into AI14 mouse line that expresses TD tomato. So if Cree is present, it will remove the stop cassette in front of the TD tomato and allow it to.
The answer is don't know. Is mono or
so
multiple synaptic?
I don't think they looked at that in the paper.
Right.
Sorry, my microphone is not very loud. I don't think they looked at that, and I don't think that I don't think that it's completely clear that it is also even restricted to synapse delivery. So I think more work needs to be done to make sure that this is actually a real anterograde tracing method. But if it works, I think it could be very useful. Yeah.
Yeah, a second question is just by curiosity. Is there any work have been done looking at AAV vectors that specifically through electrical synapses instead of chemical synapses?
I don't think so. I'm not familiar with anything.
Are there any known post-translational modifications on the capsids that you work with?
Yes, actually there are. So during production, you can get deamidation, so from an asparagine to an aspartate or isoaspartate, and that's been shown fairly recently. And just like that happens with antibodies too, and how much that actually affects the function of the virus, I think is still not determined. They probably get phosphorylated at least in vivo. Whether that happens during virus production, I don't know, but there's some reports that phosphorylation can affect their function later on as well. Yeah.
And what's the approximate volume of the capsid on the inside?
Do you know, Simone? I don't have a number for you. Enough to hold about around 5 kb of DNA, single-stranded DNA, but I don't actually know what the volume is.
My question is directed at something that Simone had mentioned. You said that when the viral capsids are self-assembling, a very high percentage of them come out empty. I was wondering, that seems sort of cumbersome and like sort of a inefficiency that you have to spin them there. I was wondering if that is just a part of the way you make these and that's the way that it's going to be, or if you see the possibility for increasing your efficiency in that step and not having to concentrate them.
Yeah, so I think increasing packaging would definitely help. And a lot of people have actually been working on that. I think it actually happens if you do that with wild-type viruses, so that's not something that's necessary due to the fact that you try to package a transgene instead of viral genome. So it's kind of still tricky. It's also going to depend a lot on the production system that you use. So some people have kind of tried to show that if you use stable cell lines, for instance, you're going to increase a little bit the amount of full particles that you get. So something people are actively working on, but they haven't been able to fully solve at the moment. So you get a lot of empty particles, just find a way to fill them.
It would certainly be a useful thing to solve because manufacturing for the gene therapy trials is a huge bottleneck and a huge problem. I mean, some of the like DMD patients are getting 1 × 10 to the 14th viral particles per kilogram. So that's a lot of virus, and that's, I don't know, a few thousand large plates of producer cells per patient, right? So it's not. There's a reason why these therapies are so expensive, and that's one component of it for sure. Yeah, so if someone solves that, that would be a potential 10-fold increase in efficiency right there.
Where are you putting the 7-mer insertions? Is that in VP3 or 1 or 2?
So it's in the VP3 part of the capsid, but that's shared amongst VP1, 2, and 3. So they all, VP1 and 2 are just longer versions of VP3. So it's in all of them. It's in a surface loop. It's in the loop 8 is what people call it. So it's in a loop domain that puts it right at one of the spikes at the 3-fold axis of symmetry.
So in terms of your species, cross-species question that you have, are there any efforts of creating like a blood-brain barrier organoid-like human iPS derived? Would that be a way to sort of test some of your questions?
I think so. So I mean, we're exploring that possibility. I think there's quite a few groups that are working on iPS-derived BBB models.
Yeah.
Some of them are 2D models where you put them on a transwell cassette and you can measure things that go through the cell, that get transcytosed through the cells and come out on the other side. There are models where you can make small spheroids where the endothelial cells actually form a tight junction layer on the outside, and you can put other types of cells inside and look for things that get in. And there are, I don't know if there's any, maybe Lee knows this, I don't know if there's anyone making organoids that actually have endothelial cells or have some kind of flow. I think that would be great. And there are some microfluidic devices as well that you can make channels in a collagen. You can basically make a collagen matrix and have a channel in it and use microfluidics to flow things through, and you can get endothelial cells to seed that channel and put other types of cells on the other side. And there's a lot of technology coming up in this space that I think will be really helpful. Yeah. Morgan.
So earlier on, you were talking about all these clinical trials going on with AAVs, and I'm just curious to get your opinion on the AAVs, AAV9 based therapies that are in trial that use systemic injection. Why do you think they would not work adequately, or do you think that they could work?
I think it completely depends on the indication, right? So for things like some of the lysosomal storage diseases, where you could deliver something and have cross-correction from having one cell express and having an enzyme be released that can then have effects on other cells, the efficiency might be high enough that you can have a real effect. I think if you wanted to, I mean, I guess a good example is, Avexis has had really good success using AV9 in neonates to treat SMA1. SMA2 usually isn't diagnosed until a little later. It's not as severe. So if you don't have an early diagnosis, then they're not going with systemic AV9 as a way of treating SMA type 2. They're already in clinical trial to use intrathecal delivery of AV9 to try to get higher doses of virus into the motor neurons in those kids. Because if you go later when they're a little bit older, the efficiency of AV9 crossing the blood-brain barrier goes down dramatically. And so it's not going to work necessarily for things like Huntington's or anything where you have to have a cell autonomous effect, I think it's less likely to be successful unless you can target a large enough fraction of the cells that you need to target. And so I think it's very indication specific.
The intrathecal AAV non-SMA doesn't correct the systemic.
Well, it might to some extent. When you inject things into the CSF, a lot of it leaks out. It's not a little, it's a lot, a large fraction. You actually get pretty good transduction of liver and some of the other organs. So,
yeah, I have a question, Ben. So, here.
There you go. Yeah, couldn't find it.
There anyone trying to use the surface lysine or cysteine to cross-link with small molecule or neuropeptide, which increase the selectivity to certain cell type?
Yeah, I think there is some work going on like that to try to use cysteines and other things, or put in some functional domain on the surface of the capsid that you could then attach other things to that. I think there is work going on in that regard, and other people are taking similar approaches where they're, rather than doing chemically, actually genetically encoding it, but putting things like DARPins or other small proteins that you can evolve to bind something through another system and then apply that into the capsid. Yeah.
Do we have any further questions? All right. Thank you very much, Ben.
Thank you.